welcome to week 10 of plant biology here at NICC. This week we're going to be stepping into the world of genetics and evolution, and to begin that conversation you guys are going to have to read in chapters 10 and 11 in the Concepts of Biology OER textbook. So again, we're going to be looking at genetics and genetic manipulation as well as basics of evolution, so chapters 10 and 11 in Concepts of Biology. Starting off, we're going to be in chapter 10, and we're really going to start out and sort of get a review of cloning and genetic engineering. So how are we putting the genetic sorts of information that we learned about last week into practice? And so we can look at these genetic processes as a series of steps to, in a way, manipulate or recreate what happens naturally in a laboratory setting and modify it so that we can utilize our technology and understanding of these systems to achieve a specific result that we might be looking for that wouldn't otherwise occur under natural conditions. To start out with, we can look at how genetic material is moved and transferred, and so we start out looking at sort of the basics of DNA and RNA and understanding the difference between those two types of nucleic acids. And the book goes through a number of different techniques that scientists use to look at DNA structure. So we can look at gel electrophoresis, so we can see how DNA tracks in different sorts of samples. We can use what's called polymerase chain reaction. PCR is one of the major technological advancements that has allowed for the rapid development of DNA technology. So spend some time and look at PCR and understand how that system works. That's an incredibly important process. They talk about cloning, so we can look at cloning specific parts of DNA and how that works. We can look at the different shape, or rather the shape of DNA and sort of the sticky ends and how they work together to form little plasmids that can act as recombinant DNA. We can look at reproductive cloning. So the most famous and first instance of reproductive cloning was a sheep named Dolly, and that was a big deal when that came out. That was in 1996 or 94. It's in the text in it, right? But that was a big deal when that happened, because that was the first time scientists had successfully cloned a full organism like a mammal for these sorts of purposes. The text then goes into looking at gene therapy, so how we can use genes to diagnose and treat genetic disorders. Not necessarily the most germane towards plant biology, but it helps build our understanding of what genetics are used for. Also looking at the production of vaccines, hormones. You talk about transgenic animals, and then they start talking about transgenic plants, which for the purposes of this class is probably the most sort of important component of this chapter. When we're looking at transgenic plants, we're really looking at plants whose DNA has been modified. A few folks have touched on GMOs in writing previously this semester, and I think this is a great time to really take a little bit of extra time so that you guys have a pretty good understanding of what a GMO, or genetically modified organism, is. And a genetically modified organism is, in the case of plants, is a plant which has DNA from another organism artificially inserted into its code. Usually these additional pieces of DNA are for desirable traits, like disease resistance or herbicide resistance or other nutritional value differences, shelf life differences. So these are all traits that have been found in other organisms that are then cut out of those organisms and moved over to this new organism. So when we're talking about genetically modified organisms, we're really talking about organisms whose entire genetic code has been added onto artificially in the laboratory section. This is not a sort of traditional plant breeding process where a trait has been found in, for example, let's say corn, for example, that one strain of corn has a desirable trait, and researchers are simply breeding that into other lines in sort of traditional ways. This is specifically where these genes, this genetic information, is being cut out of one organism and moved over into a different one. So this transgenic technology is very different than traditional plant breeding. We can look at a number of different sorts of GMO or transgenic traits that are commonly used or highly desirable in an agricultural sense. So researchers in an agricultural sense often use this agrobacterium as the sort of transformative bacteria that really allows for this DNA insertion into other plants. A very common trait that's used for agricultural crops, especially in Iowa, is what's called Bt corn, Bt beans. And this Bacillus thuringiensis is a bacteria that really is effective at killing usually things like caterpillars that feed on agricultural crops. And if plants can then produce these crystals, this Bt toxin, the plants are then able to utilize this toxin that bacteria produce and express it in their own genetics. And they then are able to sort of fight off these predators, or plant predators rather, without the use of additional pesticides. And so it's a way of introducing pesticides into, or rather a pest controlling agent into plant populations that otherwise wouldn't occur. Again, it's a way of taking genetic material from one organism, a bacteria, and moving it into a different organism altogether. And so Bt technology is very, very commonly used in the agricultural setting for a number of different crops. The chapter then goes on to talk about looking at genomes, which is the full genetic code of an organism, and understanding how scientists sort of develop these genetic maps of organisms. This is an important process when we're trying to understand how organisms function, because if you look at the DNA of an organism, we can really see what that code is telling us about what this organism is going to do. So we can look at mapping out genomes of model organisms, and then we can look at different ways of applying these genomics in an applied way rather than in just a conceptual way. So we can look at predicting risk, we can look at how genetics are associated with different DNA codes, pharmaceuticals, we can look at larger issues of genomics. We can also look at what are called biofuels. So when we're looking at the development of new biofuels, we can look and say, how do the genetics influence how plants produce the sorts of molecules that we're interested in, and then how can we modify organisms to better exploit those resources. The book then talks a little bit more about genomics in agriculture. So again, really utilizing these genetic tools to breed for desirable traits. You can look at proteomics, which is really looking at the protein codes, which is sort of secondary to genomics, which is looking at the genetic code, but also an important tool when we're looking at studying the genetics of organisms. All right, so that sort of wraps up chapter 10, and that should get you a good start looking at these concepts of genetic engineering and putting some of this genetic information that we talked about sort of last week into action in more of an applied way. All right, I'm going to add another lecture for chapter 11, and so have a great weekend, and we'll talk to you later.